This is part 1 of 3 on chapter 4, a survey of prokaryotic cells. So in this chapter, or in this unit, we're going to start to look at different organisms that are studied in microbiology. We're going to be looking at living things that have prokaryotic and eukaryotic cells, and there are 8 characteristics of life. So these are 8 things that living things do and how we actually define if something is alive or not. So the first one, living things have to reproduce, so they have to produce the next generation, and as they reproduce, they pass down their genes. So that's the heredity part. Our second characteristic, living things have to grow and develop over time, so things will grow in size and they will develop or change how they look. The third characteristic, metabolism. So all living things have to have a metabolism. They do chemical reactions. They use energy. They can create energy. So that's the metabolism part. Number 4, living things, they exhibit movement and or irritability. So this means that they can respond to their environment either by moving or by reacting to different chemicals, reacting to light, or any other type of environmental stimuli. The fifth characteristic is cell support, protection, and storage mechanisms. So we're going to be looking at different types of cells. Those cells have to be supported. Some of the cells have protection mechanisms, and a lot of cells can store energy in different forms. Number 6, living things have to transport nutrients and get rid of waste. Number 7, living things have to self-regulate, or they have to maintain what's called a homeostasis. A good example of homeostasis is the ability to regulate temperature, which mammals can do. And then the 8th characteristic is biological evolution. So things have to be able to adapt to their environment or change over time from one generation to the next. All living things are made up of cells, and cells are the smallest, most basic unit of life. So all cells we're going to look at, they exhibit those 8 characteristics that I just went through. And there's 2 different types of cells, prokaryotic and eukaryotic cells. And common characteristics to these 2 different types of cells include a basic shape. So a lot of times they're spherical, but if they have some type of cell wall to them, they can be cube-shaped or cylindrical. The second common characteristic of the 2 types of cells is the internal contents. So all cells have this kind of gelatin liquid substance in them called the cytoplasm. And that cytoplasm is surrounded by the cell membrane or the plasma membrane. The third characteristic that all cells share is that they have DNA in the form of a chromosome or chromosomes. They also have ribosomes, which synthesize proteins based on the DNA. And then they also have metabolic capabilities. This slide is showing the 2 different types of cells and kind of some basic characteristics of them. On the left-hand side, we have a cell called a prokaryotic cell. Prokaryotic cells, they do not have a nucleus to them, which means that their chromosome or their DNA is just kind of floating in this chromosome region inside of it. So their DNA is not protected in any way. Besides having a chromosome, which is usually circular in shape, they also have ribosomes, which again synthesize proteins based on the DNA or that genetic blueprint. Prokaryotic cells, they also have a submembrane, and that defines what's inside the cell versus what's outside the cell. And then outside of the cell membrane, you can have a cell wall, or you can have flagella, or lots of different types of things that we'll go through. On the right-hand side, we have a eukaryotic cell. Eukaryotic cells, they do have a nucleus, and that's the purple circle in the middle of your eukaryotic cell up on the slide. So this nucleus, this is where the DNA is found, and the DNA is usually in the form of chromosomes that are linear, so you can have multiple chromosomes inside of the nucleus. In addition to the nucleus, eukaryotic cells also have ribosomes, which again synthesize proteins. And then you can have other types of organelles. For example, there's mitochondria, you can have chloroplast, there's endoplasmic reticulum, Golgi apparatus, and we'll get to all the different types of organelles. You can also have your cell membrane, and again that defines what's inside the cell versus what's outside the cell. And then on this cell, this eukaryotic cell on the slide, they have a flagellum. And we'll get to the function of that when we get to eukaryotic cells. In this chapter, we're really going to focus on prokaryotic cells, the cells that do not have a nucleus, they do not have organelles. So here's kind of the basic structure of a prokaryotic cell. And you're going to have to know all of the different parts that are labeled up on the slide. So you'll need to know the names of the parts and also their function or what they actually do for that prokaryotic cell. And we're going to be looking specifically at a bacteria cell as our kind of model for our prokaryotic cell. So we're going to start off looking at the structures that are outside of the plasma membrane, and then in the next part of this chapter we're going to go inside the plasma membrane and look at the rest of the structures that are in there. Here are the different external structures. So again, these are structures of the prokaryotic cell that are found outside of your cell membrane or outside of the plasma membrane. So one thing is that you can have appendages. So these are structures that kind of stick out, kind of like arms and legs for the cell. Some appendages are used for motility. These include the flagella, axial filaments, and we'll look at what's the periplasmic flagella. So those are used for motility or to help that cell move through the environment. Other appendages are used for attachment to different surfaces, or they can function as channels to exchange genetic information. And these are going to include the Fimbriae and the Pili. In addition to the different types of appendages, you can have a Glycocalyx, which is a surface coating. So we'll look at the appendages first. So the flagella, it comes in, or it's made up of three parts. So you have the filament part of the flagella. This is a really long, thin helical structure. It's composed of a protein called flagellin. So this is kind of, the filament part is the long part that actually helps the cell move. This filament, it goes into a part called the hook, and you can see this on the diagram. And then that hook, that is the part that attaches the filament to the basal body. And the basal body is a stack of protein rings that is anchored into the cell wall of your bacteria cell. Some bacteria cells only have one flagella, and they're called monotricious. That's in the upper left-hand side. So that's a bacteria cell that has one flagella. If you have multiple flagella coming out both sides of your bacteria cell, it's called amphitricious. Then if we go to the bottom, if all of your flagella just come out of one side of your bacteria cell, it's lophotrocious. If all of your flagella just are randomly coming off your bacteria cell, it's peritricious. So there's different types of flagellar arrangements, and we can use this to actually identify different species of bacteria. So there's a flagella stain. It's a structural stain that we talked about in chapter 3. That flagella stain, it's used to see the flagella and to identify their arrangement. In prokaryotic cells, these flagella that are used for movement in the environment, they will whip around in a 360-degree fashion. So they're actually rotating in a circle, and that's going to propel the bacteria cell forward. If the bacteria cell has multiple flagella, as shown with the purple bacteria down here, when the bacteria wants to move forward, those flagella will wrap around each other. They'll spin around in 360 degrees. And then when the bacteria cell wants to stop, the flagella will come apart. They'll be individual, and then they'll just rotate where they are. So it kind of can stop the movement with that. These flagella, they have bacteria cells move through the environment. And usually bacteria cells are trying to respond to some type of external stimuli. And this can be things like a chemical stimuli, and this is called chemotaxis. If the bacteria cell is moving towards the chemical, it's called positive chemotaxis. If the bacteria cell is trying to get away from the chemical, it's called negative chemotaxis. Same thing with light. So bacteria cells that move towards light, it's called positive phototaxis, because photo means light. If a bacteria cell is trying to get away from light, it's called negative phototaxis. They're trying to get away from that stimulus. So here, if your bacteria cell has no attractant or repellent, the bacteria cell just kind of randomly moves in its environment. If the bacteria cell is moving towards some type of chemical, the movement's going to kind of be random, but you're going to have a general direction that it's going to be moving. A special type of flagella called the Periplasmic Flagella. This is an internal flagella, so it's not actually sticking out. It doesn't look like an appendage like a normal flagella would. So these internal flagella, they're found enclosed between the outer sheath and the cell wall of your bacteria cell. So these flagella, instead of whipping around in a 360 circular fashion, these flagella instead, they're going to contract kind of like a muscle. This is going to cause your bacteria cell to twist or to flex. And bacteria cells, the spirillum bacteria cells, the ones that look like a spiral, a lot of times they have these periplasmic flagella, and they'll move by twisting or turning. So that's how they move, using these. Another type of appendage is the Fimbriae. So Fimbriae, they're really fine, short, hair-like structures that can cover the whole cell, and they function in adhesion. So this is when a bacteria cell wants to stick to a surface, or the bacteria cells want to stick to each other. So they're going to adhere to the other cells or to the surface of, like, the table, for example. So picture A up at the top. This shows bacteria cells, so the orange, blue, rod-shaped bacteria. And then the greenish-yellow hair-like structures, those are the Fimbriae. The picture in B, this is showing E. coli cells, and they're attaching to intestinal cells in some type of animal. So these E. coli cells are going to anchor themselves into the intestinal wall. Another type of appendage is called the Pili. These are longer tubular structures. They're made of a protein called pilinin. They're found in gram-negative cells, and we'll get to the different types of cell wall in a little bit. In Pili, their function is in conjugation. Conjugation is a form of, kind of a form of reproduction, but this is where two bacteria cells, they can swap DNA through this pili. It's sometimes called the sex pili. So conjugation, it's just one way that bacteria cells can share genetic information, kind of mix up their genes. So far we've looked at the different types of appendages. We looked at the flagella, the periplasmic flagella, and then we also looked at the Fimbriae and the Pili. Remember, these are different types of appendages. They're found outside of your cell membrane. The other external structure we're going to look at is called the glycocalyx. The glycocalyx is a coating of molecules that's external to the cell wall, and it's made up of sugars and or proteins. If the glycocalyx contains a lot of sugars, this forms more of like a slime layer. So it's really loosely organized and attached together. It's usually sticky because it's made up of sugar. If your glycocalyx is more made up of proteins, it's usually a little harder. It's called a capsule. It's very highly organized. The molecules are tightly attached to each other, and it forms more of a hard layer. So you can have different types of glycocalyx, kind of the sticky slime layer or the harder capsule glycocalyx. So the function of the glycocalyx, so there's a few different functions. It can help protect the cell from dehydration and nutrient loss, so losing water or losing food. The glycocalyx, it can inhibit killing by the white blood cells by a process called phagocytosis, and this contributes to the bacteria's pathogenicity. So to kind of explain this a little more, if a bacteria cell has a capsule, this makes it harder for our white blood cells to capture the bacteria cell and to destroy that bacteria cell. So that means our immune system can't work as well when bacteria cells have a capsule to them. Another function of the glycocalyx is for attachment. So this is, again, attaching to different surfaces like the table or to the intestinal wall. And specifically, the glycocalyx helps in the formation of something called a biofilm. And I'll show you what that is on the next slide. But here we have images. We have different colonies of bacteria. So colonies without a capsule, they look more kind of spread out. Whereas bacteria colonies that have a capsule, they have a little more structure to them. Those cells can stick together and form this perfectly round colony. The other picture on the right, this is showing a stain, a capsule stain, and we'll most likely be doing a capsule stain in lab, so you get to actually do one of those. And that's another structural stain, so it's helping us identify a certain structure for that bacteria. So here is an example of a biofilm on a catheter. So these biofilms, if the catheter is not clean, it can lead to a urinary tract infection. So this is why they really stress having clean catheters, otherwise you're going to cause that person to be in a lot of pain. So on the catheter, or any type of surface, you're going to have what's called a biofilm form. And biofilm, it can be composed of bacteria cells. So we have a Staphylococcus species of bacteria on this catheter. And it can also include fungal cells, so other types of microorganisms. And a biofilm is basically just a bunch of different microorganisms sticking together, forming this film. Another really good example of a biofilm is the plaque that builds up on our teeth. So a plaque is just a bunch of bacteria waste, sugar, possibly some fungal cells forming this layer. So far we've looked at the appendages on our bacteria cell, our prokaryotic cell. So next we're going to go into what's called the cellular envelope. So the cell envelope, this is the external covering outside of the cytoplasm, and it consists of two layers. So it includes the cell wall, and it also includes the cell membrane. So the cell wall is actually outside of your cell membrane, but they work together in this cell envelope. So the cell wall is really, really important. So the cell wall actually contributes to we having two different groups of bacteria demonstrated by a stain called the Gram stain. And the Gram stain is a type of differential stain that we looked at in Chapter 3. So your cell wall, you can either have gram-positive cell wall bacteria. This is where the cell wall is really thick, and it's composed primarily of peptidoglycan. This is a protein carbohydrate molecule. And then in addition to this really thick peptidoglycan layer, you also have your cell membrane associated with it. A gram-negative cell wall, this is where you have the outer cell membrane, there's a thin peptidoglycan layer, and then you have your normal cell membrane. So there's two different types of cell walls, and they're called gram-positive and gram-negative. These cell walls, they help to determine the shape of the cell, and we'll get into the different shapes that we'll see in lab. In addition to creating or giving the cell a shape, the cell wall also prevents something called lysis. And lysis basically is just bursting open of the cell due to osmotic pressure or due to changes in water flow or osmosis in and out of the cell. Cell walls, they're primarily composed of the molecule peptidoglycan. And again, peptidoglycan, peptide is protein, glycan means carbohydrate. So it's kind of this combination of protein, so a bunch of amino acids hooked together, and they're also associated with some type of carbohydrate. And this helps make up that cell wall. And like I just mentioned, a gram-positive cell wall has a really thick peptidoglycan layer. And it's about 20 to 80 nanometers thick. So nanometers very, very tiny, but it's pretty thick compared to the gram-negative cell wall layer. In these gram-positive cell walls, some of these cells have that periplasmic space, and that's where you're going to find the periplasmic flagella. And that is between the cell membrane and the cell wall. So it's kind of this open space. You can have the periplasmic flagella, and then those bacteria cells can move by twisting. The gram-negative cell wall, they have an inner and outer cell membrane, and then their periplasmic space contains a really thin peptidoglycan layer. The very outside of the gram-negative cell wall, it's made up of lipopolysaccharides, or LPS. These are a combination of a lipid and a polysaccharide, or a really complex carbohydrate. And we'll be doing the gram stain in lab, so we'll be reviewing this quite a bit, the difference between gram-positive and gram-negative cell walls. This slide is showing another way to look at the two different types of cell walls. On the left-hand side, we have a gram-positive cell wall, so you can see the peptidoglycan layer is very, very thick. And then under that peptidoglycan layer, you have your cell membrane at the bottom. On the right-hand side, we have our gram-negative cell wall shown here. So we start with a membrane layer on the very outside, and then under that outside plasma membrane layer, you have the really thin peptidoglycan layer. And then under the really thin peptidoglycan layer, you have your cell membrane under that. So your gram-negative cell wall, it's made more of the lipopolysaccharides, so the lipids and the sugar, whereas your gram-positive cell wall is made more of your proteins and carbohydrate molecules. So these are your two different types of cell walls. So in order to look at the two different types of cell walls, or to distinguish them or identify them, we do a differential stain called the gram stain. And you're going to be doing the gram stain pretty much every single time in lab for the first 8 or even 9 weeks of lab. So the gram stain is very, very important. So it's good to understand how this works. And we'll go through it in lab again. But your gram-positive cell wall, it's going to retain a stain called crystal violet. And that's going to make them look purple when you look at them in the microscope. Whereas your gram-negative bacteria, they're going to lose that crystal violet. It gets washed off by alcohol. And we're going to counter-stain it with a red stain called safranin. And again, this is your counter stain. So we still have to stain these cells in order to see them, and they end up being red or pinkish in color. So this gram stain, we use it to identify different bacteria, and we use it to classify bacteria into different groups. And every time, or pretty much every time, any time that you collect a sample from your patient, whether if it's a human or animal, they're going to be doing a gram stain on that sample. So it's a really practical aid, very easy to do, takes maybe 5 minutes, and you can see if you have gram-positive or gram-negative bacteria. It helps you to diagnose infection. It also helps to guide with drug treatment. So there's different antibiotics. Some antibiotics only work on gram-positive bacteria. Other antibiotics only work on gram-negative bacteria. So it's really useful to kind of know what type of infection you're dealing with, so you can actually start to treat it almost immediately. In the gram stain, it has a few steps to it, so it's a little more complicated of a stain. So the first step is that you stain everything with crystal violet. And crystal violet is the primary dye. It's going to make everything look purple. And then on top of that crystal violet, you're going to put grams iodine, which is called a mordant. And this is going to help that crystal violet get into your gram-positive bacteria cell walls. The gram-negative cell wall, your crystal violet's going to be washed off with alcohol. It's a decolorizer. So if you look on the chart, by the third step, when you wash it with alcohol, your gram-positive bacteria, they're still going to be purple because that dye has been trapped inside of the cell wall. Your gram-negative cell, the crystal violet is going to be washed off, so it's going to look clear. And we can't see clear cells very well with the microscope, so we have to use a counter stain to make it look pink or red. And that's the safranin, that's your counter stain. So this is how you're going to get the gram-positive being purple, gram-negative, it's going to be pink in color. Most bacteria either have a gram-positive or a gram-negative cell wall. But there are a few different species where they have a non-typical cell wall, so they're kind of out of this gram-negative, gram-positive. So these include Mycobacterium. That's a really good example. So these bacteria, they have a gram-positive cell wall structure, but they have something called mycolic acid included into their cell wall. So this allows these bacteria to have a really high degree of pathogenicity, so they can cause very, very bad infections. Mycobacterium, one species, causes tuberculosis. In addition to high pathogenicity, the mycolic acid allows these bacteria to have a really high degree of resistance to certain chemicals and dyes. So it's really hard to kill these bacteria. So this mycolic acid that allows these cells to cause these horrible diseases like tuberculosis that I mentioned, it's the basis for the acid-fast stain. And we're going to be doing the acid-fast stain in lab. It's another type of differential stain used to identify acid-fast bacteria. And then finally, some bacteria can just have no cell walls. So Mycoplasma bacteria, their cell wall is stabilized by sterol, so a type of lipid. And these cells are pleomorphic, so they actually change shape because they don't have this rigid cell wall to them. So far we spent a lot of time talking about the cell wall, which makes up part of the cell envelope. The other part of the cell envelope is the cell membrane. So I have one slide looking at cell membrane structure. So cell membrane, it's a phospholipid bilayer, and so that means 2 layers of phospholipids. On the phospholipids, remember they're amphipathic molecules, so the head of the phospholipid is hydrophilic, so it's going to face out towards either the external environment or in towards the internal environment of the cell. The other part of the phospholipid is hydrophobic. Those are those fatty acid tails. They're going to face in towards each other. So bilayer, you have 2 layers of phospholipids, fatty acid tails facing in. In these phospholipids, you also have embedded proteins. And this creates something called the fluid mosaic model. So your cell membrane or plasma membrane, it's fluid. These phospholipids and some of these proteins can move around. They're not stationary. It's also considered to be a mosaic, and that just means that it's made up of lots of different components. And you can see that on the slide here. So you have your phospholipids, you have different types of embedded proteins. So there's integral proteins is one example. That's the big green one. There's peripheral proteins. You also have glycolipids, so you have lipids that have carbohydrates attached to them. So you have this mosaic of different molecules making up your membrane. So your cell membrane, the main functions, it provides a site for energy reactions in your prokaryotic cell. It also allows for nutrient processing and synthesizing different products the cell may need. In addition to that, the cell membrane is also selectively permeable. So this means that certain things can move in and out of the cell. It can allow food to come into the cell, and it can allow waste to go out of the cell. And selectively permeable means that certain things can move across, other types of molecules cannot move across. So this is your cell envelope. You have your cell membrane, and then you can have different types of cell walls that go along with your membrane.